Today we are going to discuss the organization of human body at cell level. Let's see the introduction part. All living things are made up of cells. Cells are the smallest working unit of all living things. That means if we take any all living things or all living animals, so they are mainly made up of cells. So a cell is defined as the smallest basic unit of life that is responsible for all of life's process. So our body's any function has done by the cell only, or cell is the structural, functional and biological unit of life. Cells they are known as building blocks of life. The first cells were observed and named by Robert Hooke in 1665 from slice of work. That means in 1665 Robert Hooke is the scientist who has observed the cells, which is the smallest working unit, and has given the name as a cell. Then as a continuation, the size of cell can be vary from minimum of 6 micrometron, that of the resting lymphocyte, to a maximum micrometron, that of a mature ovum. That means if we check the size of lymphocyte at the resting period, their size will be 6 micrometron, that is a minimum, and the mature ovum size, that is the maximum. Mature ovum means the ovum which is secreted during the ovulation from the female body. The study of cell is called cytology, and the study of tissue is called histology. Characteristics of human cell. So it is the smallest unit of body. It is binding block of life. Cell become divided except the neuron cell. That means all the human cell, they can divide and they can make daughter cell except neuron cell. Number of cell, that is from 100 to 14 trillion approx. Size of cell, that is from 1 to 100 micrometron. Structure and functional unit of living organism. So cell structure. So an eukaryotic cell consists of the following structure. So human cells, they are the eukaryotic cell. They are mainly present, the major structure present as in a cell are cell membrane, nucleus, cytoplasm and its organelles. Cell membrane is the outer covering of the cell. Nucleus, mainly they are present at the near or center of the cell, and the cytoplasm, which is a fluid present inside the cell, and other organelles. Let's see the cell membrane or the plasma membrane. Cell membrane is a protective sheet that envelops the cell body. So another name of the cell membrane is plasma membrane, which are protected the cell from the outer force. They act as an envelope. It separates the fluid outside the cell called extracellular fluid and the fluid inside the cell called intracellular fluid. That means this membrane, they are separate the ICF and ECF. It is semi-permeable membrane and allows free exchange of certain substance between the ECF and ICF. So this plasma membrane, they made in a such a way that they are semi-permeable and they are allows the free exchange of certain substance which are needed for the cell and composition of cell membrane. There are mainly three types of substance are present inside the cell membrane. They are protein 55%, lipid 40%, carbohydrate 5%. In a picture, so we can see that they are mainly protein which are embedded inside the membrane, and the phospholipid, they are present in the outer and inner layer of the cell. Then carbohydrate, they are mainly present above the lipids. The cell membrane is fluid mosaic model. In the cell membrane, inside the cell membrane, we can see the fluid mosaic model. So because of that fluid, molecules in the cell membranes are constantly moving and changing. Nucleus. The cells with nucleus are called eukaryotes, and those without nucleus are known as prokaryotes. The cells which are without nucleus, that are called prokaryotes. They are the and the another one, that is eukaryotes. Eukaryotic cells are the human cell or the advanced developed animals which are their cells are the eukaryotic cells which are with nucleus. Most of the cells have one nucleus, that is called eunucleated. Then few types of cells like skeletal muscles cells have many nuclei, multinuclear. That means in the skeletal muscles the number of the nucleus are more than the one. Generally the nucleus is located near the center of the cell and it is mostly spherical in shape. So if we see the nuclear component, there are mainly three components: nuclear membrane, nucleoplasm and nucleolus. So nuclear component, under that nuclear membrane, the nucleus is covered by a double layer membrane called nuclear membrane. So the nucleus has covered by the nuclear membrane. Next is a nucleoplasm. It is a gel-like ground substance and contain large quantities of genetic material in the form of DNA. That means inside the nucleoplasm the DNA are occupied in that state. DNA substance are present, and DNA is made of the chromatin thread, and DNA is mainly made up of the chromatin thread, so which is become chromosome during the cell division. Nucleoli, one or more nuclei are present in each nucleus. So in one nucleus, one or more than one nucleoli may be present, and the nucleolus contain RNA and some proteins which are similar to those found in the ribosome. The nucleus which are contain RNA and some proteins which are similar to those found in the ribosome. So function of the nucleus, mainly they control of all the activities of the cell. So mainly the cells, the cell division and all, they are controlled by the nucleus. Synthesize RNA, they are producing the RNA, and sends genetic instruction to the cytoplasm for protein synthesis through messenger RNA. So from mother cell to daughter cell, or either from the in the meiosis process, from mother cell or from parent cell to daughter cell, they are sending the genetic instruction through messenger RNA. Cytoplasm is the fluid present inside the cell, and it contains a clear liquid portion called cystosol, which contain various substances such as protein, carbohydrate, lipid and electrolyte. Here also you can see that the fluid inside the cell, we can see the cytoplasm. Next, cell organelle. We have seen that apart from the nucleus, the cytoplasm of typical cell contain various structures that are varied to as a organelle. So cytoplasm, and if we accept the cytoplasm and the nucleus, the other cell organelle present in the cell, they include endoplasmic reticulum, then ribosome, mitochondria, Golgi complex and various types of vesicles. The cystosol also contains cytoskeleton made up of microfilament and intermediate filament and microtubules. So here in the right side of the table, we can see that cell organelle we have divided in two parts: membrane bounded cell organelle and non-membrane cell organelle. Here I have listed some points which are the organelle which are bounded with the membrane. The another side, that is the organelle which are without membrane. So under the membrane bounded cell organelle, the first one is the endoplasmic reticulum, then Golgi apparatus, then mitochondria. Membrane bounded vesicle including phagosome, lysosome, peroxisome and exocytic vesicles. And under the non-membrane and cell organelle, cytoskeletal including microfilament, microtubule, intermediate filament and ribosome. So endoplasmic, first we discussed the endoplasmic reticulum. Endoplasmic reticulum is made up of membranous tubule present within the cytoplasm of eukaryotic cell. In the eukaryotic cell, we can see that the endoplasmic reticulum, they are present in the they are mainly made up of membranous tubule and present as a arenes, and this structure form a interconnected network which act as a link between the organelles and cell membrane. So mainly they have they made the connection between the organelle, other organelles and cell membrane. The endoplasmic reticulum is two types of, namely. So mainly these endoplasmic reticulum, they have divided in two types. One is rough endoplasmic reticulum, another is smooth endoplasmic reticulum. The rough endoplasmic reticulum mainly it is one of the reticulum which is which the granular ribosome is attached, and this gives the rough appearance, so it is called rough endoplasmic reticulum. The endoplasmic reticulum which are attached with the granular ribosome, that are called endoplasmic reticulum, and another one that is smooth endoplasmic reticulum. Smooth endoplasmic reticulum is also called granular endoplasmic reticulum because of its smooth appearance without the attachment of ribosome. As the endoplasmic reticulum is without ribosome, they are smooth in appearance, so they are called smooth endoplasmic reticulum. So if you see the function of the endoplasmic reticulum in the picture, also you can see that the smooth one they are without ribosome, and the rough one they are with ribosome. So if we see the function of the endoplasmic reticulum, rough or granular endoplasmic reticulum is concerned with the synthesis of protein. So mainly the rough endoplasmic reticulum, they are involved in the synthesis of protein to produce protein, and a smooth granular endoplasmic reticulum lacks ribosome and it's concerned with the synthesis of lipid, steroid, cholesterol and carbohydrate. So mainly they as they are the ribosome are lacking in the smooth endoplasmic reticulum, they are mainly concerned with the synthesis of lipid, steroid, cholesterol and carbohydrate. Next come to the Golgi apparatus. This is the another cell organelle. Golgi apparatus, another name is Golgi body or Golgi complex, is present in all the cell except RBCs. So Golgi apparatus it is present in all type of cell except RBC. RBC is the red blood cell, that is the one type of blood cell. It consists of 5 to 8 flattened membranous sac called cristae, and the Golgi apparatus is situated near the nucleus. It has two ends or faces, namely cis face and trans face. So it has two types of face, cis face and trans face. The cis face position near to the endoplasmic reticulum, and the reticular vesicle from endoplasmic reticulum enter the Golgi apparatus through cis face, and trans face is situated near the cell membrane, and the processed substance make their exit from the Golgi apparatus through trans face. So if any substance need to transfer to the cell, so it will come first near to the trans face, then it will through the Golgi apparatus it will enter, and through cis face it will enter the inside the cell. Function of the Golgi apparatus, it is concerned with the processing and delivery of substance such as protein and lipid to different parts of cell. So mainly they are helping in the processing and delivery of the substance. So mainly they are helping in the delivery substance such as protein and lipids to the different parts of the cell. So next cell organ, that is mitochondria. So we know that the mitochondria are called the powerhouse of cell. So mainly they are making the power or energy maker, that's why they call powerhouse of the cell. So length of the mitochondria is 5 to 12 micrometer, and the diameter is 0.5 to 1 micrometer. So component of mitochondria are: the structure comprises an outer membrane and inner membrane, and the gel-like material called the matrix. In the picture also we can see that the inner membrane, they have it consists of inner membrane and outer membrane, and the matrix which are present inside the mitochondria. The outer membrane and the inner membrane are made of proteins and phospholipid layers separated by the intermembranous space, that is cristae. It has many folds that form a layer structure called cristae. So here in the picture also you can see that the cristae is the layers, many folded layer, and the cristae and the protein of the inner membrane aided in the production of ATP molecules. ATP means adenosine triphosphate. So produce 90%, mitochondria produce 90% of the cell energy, that's why it is called powerhouse of the cell, and convert nutrient into the ATP in the presence of oxygen, that is called aerobic respiration. So RBC blood cell, that is 0 mitochondria, whereas liver cell muscles all 100 to 2000 mitochondria. That means in the RBC, as we have said earlier, that in the RBC there is no mitochondria, whereas in the liver cell we can find 100 to 2000 mitochondria in one cell. So mitochondria have several unique features among other cell organelles. they can move, change and their size, shape and divide it within the cell. So other organelles they cannot change their size and shape, but the mitochondria they can change their own size and shape, as well as they can divide within the inside the cell. Sperm do not contribute mitochondria to the zygote, only ovum contribute. That means when a zygote has formed, so normally we know that the zygote has formed with sperm and ovum. When the sperm and ovum sperm and ovum and they together they made the zygote. So in they will form one cell that is called zygote. That in that cell mitochondria will be present, but it will come from the ovum, which is come from the female or the mother cell, and another one the sperm which is come from the fathers. In that mitochondria will be absent. So membrane and bound vesicles. The cytoplasm of cell may contain several types of vesicles. Vesicles are small cellular contents. So mainly they are cytoplasm. They have mainly several types of vesicles. Vesicle means there is a small cellular container. We cannot consider there as a cell. So they perform a variety of functions. This vesicle they have different types of functions, such as helping to transfer material that an organism needs to survive and recycle waste material. So inside the cell there are so many work has done for transportation of the substance, for the recycle of the waste material, that has done by the vesicles. Other functions such as vesicles can include absorbing and destroying toxic substance and pathogen to prevent cell damage and infection. When any foreign particle try to enter in the body, they will absorb that particle and destroy toxic substance and pathogen to prevent the cell damage and infection. Vesicles also allow exchange of membrane between the parts of the cell. So these vesicles they are allow the exchange of membrane between the different parts of the cell. And membrane bound vesicle include phagosome, lysosome, peroxisome and exocytic vesicle. These are the vesicles which we have found in the cell, inside the cell. Next, cytoskeleton. From the word we can get some idea that skeletal, that means they are the framework, they give the strength. The cytoskeleton, the skeleton which is present inside the cell. The cytoskeleton of the cell is a complex network that gives shape, support and stability to the cell. It also essential for the cellular movement and the response of the cell to external stimuli. That means these skeleton they help the cell to give shape and also help in the movement and the response of the cell to external. If there is a force or something from the external, they stimulate the cell, they will help in the to respond that one. And the cytoskeleton consists of three major protein components. They are microtubules, intermediate filament and microfilament. So here we can see in the table the microtubules, microfilament and intermediate filament. Under the microtubule, the large contractile protein fiber, that means they are helping in the contraction. So mainly they are protein fiber, they are in the large in shape. They are made up of tubulin, alpha tubulin and beta tubulin subunit, and involved in the movement of organelles within the cell. They help in the inside the cell. When the organelle they want to move, them this microtubule they are helping. And chromosome during cell division. During the cell division, the chromosome has to be divided. In that also the microtubules they are helping. And cell extension like cilia and then villi, these are the part. Then microfilament, smallest protein fibers. So mainly we compare the if we compare the cytoskeleton, the microfilament is the smallest protein fibers. So mainly they are mainly with the protein. So made up of two actin strands coiled on each other. Two strands are there and they are coiled to each other and provide structural as they are coiled to each other, and they are giving support to the structure and maintain shape of the cell. They also help to maintain the shape of the cell. Helps in muscle contraction. When it is present in the muscles, they help in the muscle contraction. The last one, that is intermediate filament, thicker than the microfilament. So they are thicker than the microfilament and made up of fibrous protein. This is mainly made with the fibrous protein and help maintain the shape of cell and stabilize the position of organelle as nucleus. Sorry. Then next, ribosome. Ribosome, I think we have heard the name ribosome many times. So what is ribosome? It is mainly made up of protein and rRNA. Ribosomal RNA. Protein factories for cell. So ribosome is considered as a protein factory, as they help in the they produce the protein. Then join amino acid to make protein through protein synthesis. So they will join with the amino acid and they produce protein. So mainly ribosome are it can be attached to the rough endoplasmic reticulum and be free from the unattached in the cytoplasm. So ribosome can be two types. One will attach to the endoplasmic, that is called rough endoplasmic reticulum. Another one which will be free inside the cell. And so application and implication in the nursing, if we see that part, so genetic defect can lead to the absence of specific acid hydrolysis that are normally present in the lysosome. If there is genetic defect in the lysosome, it can affect the specific acid hydrolysis, and as a result, some molecules cannot be degraded and accumulated in the lysosome. Example, such disorders are lysosomal glycogen storage disease. So as cell extension, from the word we can say that extension means the extended part from the structure of the cell. Many cells show projection from the cell surface. There are some cells they have the projection from the cell surface. The various types of projection are described below. Cilia. So this can be seen with the light microscope, and minute hair-like projection from the free surface of some epithelial cells. In the living animal, cilia can be seen to be the motile. So the cilia we can see in the they are attached with the they are the extended part of the cell. So we can see in the living animal and they are motile. That means they can move. And mainly the cilia, they can be their hair-like projection. Functional signification of cilia: the cilia lining and epithelium surface move in coordination with the one another to total effect being that like a wave, and as a result, liquid, mucus and small solid object lying on the epithelium can be caused to move in a specific direction. A movement of cilia lining the respiratory epithelium helps to move secretion in the trachea and bronchi towards the pharynx. So as the respiratory system, for example, the respiratory system, they have the cilia, so they help the movement of the air which is entering through respiratory system, and it will move to the trachea and from bronchi towards the pharynx. The next one is flagella. These are the somewhat larger process having the same basic structure as cilia. Their structure is almost same with the cilia, and in the human body, the best example of flagellum is the tail of the spermatozoon. That means this is the tail of the spermatozoon, which is the smallest cell, and the movement of the flagella are different from those of cilia. In a flagella, movement start at its base. And microvilli are finger-like projection from the cell surface that can be seen by in microscope. Microvillus consist of an outer covering of plasma membrane and a cytoplasmic core in which there are numerous microfilament actin filament. So microvilli, they are mainly present in the inner mucous layer of the small intestine. So they are helping in the peristalsis movement. So cell movement of the cilia and flagella we can see in the picture. So they have the cell extension part. Thank you.